Hi, I'm Alini.
Hi, I'm Jessica.
I'm Chris Sarah.
I'm Madeline,
I'm Marissa.
and today we'll be talking about our project, an innovation called Lizard, which stands for Limb Integration, Zoning, and Regeneration Device, a mechanism that allows human limb regeneration. The Lizard device acts as a zone in which this process is undergone. You may be wondering, how did we get the inspiration for the idea of human limb regeneration? As you can see, there's an array of species that demonstrate the ability to regenerate limbs in other parts of the body. Some of these species, like the planaria, the hydra, and starfish, can even regrow vital organs. Now, how do they do this? Let's focus on one animal in particular. This is the axolotl, a type of salamander that is able to regenerate its legs. It has been found that an essential element for this behavior are its cells, which interested us greatly and sprang forward our research into how we could copy this ability in human beings.
If animals are not your cup of tea, another inspiration we had comes from the Marvel Universe, which has been taking over pop culture on the screen, stage, animations, and also video games. Popular heroes such as Deadpool and Wolverine are known for their regenerative healing factors that allows them to heal lost tissues at superhuman speeds. But you may be thinking, superheroes are only works of fantasy, and there's no such thing as science to back up their stories. But, well, that's not completely true. Let's take the supervillain Lizard as an example. Although his attempt on recovering his lost arm was a little too successful, Lizard was inspired by the actual animal and its ability to regrow its tail that allows the supervillain to have a strong healing factor. So, as you could probably tell, the idea of regeneration has already been circulating in the media for quite a while now.
So, what will it take to mimic these fantastic regenerative abilities? Well, as we all know, humans are capable of regenerating to an extent. However, the human body can't regenerate things like bones naturally. For this reason, issues surrounding amputation are more prevalent than you may realize.
There are nearly 2 million people living with limb loss in the United States. The main causes of limb loss are vascular diseases like diabetes, which represent 53%, trauma, which represents 45%, and cancer, which is less than 2%. The Centers for Disease Control and Prevention estimate that 33% of Americans will have diabetes by the year 2050, so the number of amputees in the U.S. is also predicted to go up. Around 500 amputations happen per day in the U.S., which adds up to around 185,000 per year. And in 2009, hospital costs associated with amputation totaled more than $8.3 billion. For people who are missing limbs in their present, their only option is to get artificial limbs or prosthetics. Our goal is to create another option for them, utilizing stem cells.
Okay, so this is what we researched. Stem cells are able to self-replicate like any other type of cell, so they're undifferentiated cells. They can divide into more cells or differentiate into cells with their own functions. Unlike other cells, they go through apoptosis or cell suicide to avoid overgrowth and cancer. So there are three main methods to extract stem cells, which include bone marrow extraction from a drilled hole, usually in the pelvis, lipid cells from liposuction, and blood extraction through apheresis, where blood is taken from the patient, the stem cells are extracted from the blood, and the blood is put back into the patient.
To more effectively use these stem cells, however, they need a map to the body part that needs to be regenerated. This is where the Hox gene will come in. The Hox gene is responsible for assigning the basic characteristics of a species. For example, in flies, it would assign 2 wings, 2 antennas, etc. By using the Hox gene, the stem cells will be directed to the appropriate part of the limb that is supposed to regrow. Furthermore, to aid in the process of regeneration, we will also use other genes in the body. A gene known as Lin28 is already used by the body to heal itself when there is tissue damage. However, since an entire limb worth of skin will need to be created, we will amplify the effects of the gene. This will be done through gene modification of the existing Lin28. In addition, the FOXA gene in humans will be modified to mimic the FOXA gene in planaria, which is responsible for directing stem cells to the part of the body that needs to be regenerated. If the FOXA gene in humans is properly modified, it can support the stem cells in the regenerative process. But stem cells and genes are not all we need for this regeneration process.
Microbots are going to be an essential part of the humans regenerating limbs. As you can see on this microbot diagram, utilizing a micromotor, there are designs from scientists showing how microtechnology could function. In this case, the motor would be delivering growth factors to aid in the differentiation process of stem cells along with the genes. Growth factors are signaling molecules known to aid in cell differentiation. As you can see on this diagram, growth factors, along with chemical cues, help the stem cells to differentiate into different types of cells and tissues needed for the new limb.
So one of the functions lizard will have is control over the process of regeneration in the case of error. This will be done through the use of magnets. So the machine will function like an MRI, and it will use electromagnetic fields to interact with the microbots, except it will be smaller and more precise.
Similar to the way different organelles within cells are stained to be observed by scientists, the microbots in stem cells will be stained prior to the process so that it can be observed by doctors through a light microscope embedded within the machine, as you can see here on our prototype.
Using all these components, we have created a step-by-step process to regenerate a limb using the lizard device.
So the first step is to take stem cells from the bloodstream through apheresis. As I mentioned before, this will take 1 to 3 days to do, but a patient can go home each night and it's painless, which is why we're using this method.
Then the site of the amputation is inserted into the lizard device. Lizard is mobile in a way that it can be adjusted to the type of amputation that the machine is being used for.
In this step, to allow the stem cells to grow backbones and other body-like characteristics, the foxide gene will be modified to mimic a Panaria's foxide gene.
The machine injects the stem cells into the site of limb growth. The microbots are then injected into the bloodstream and target the stem cells, leading to the differentiation process.
So with a technology capable of regenerating limbs, a new option will be available for amputees. This also will be the foundation for other regeneration projects that are even more complicated, such as brain regeneration, because components of the regeneration process will already be researched. Even further, a muscle memory imprint component can be incorporated in the future.
We have some discussion questions to consider as well. As we begin to modify our own bodies, we must ask ourselves what should be left to nature and what should be in our control. Is it our natural body that makes us human, and if so, by regenerating parts of it, do we cease to be human in a way? What will be the recovery after the regrowth of a limb be like?
Although there are still a lot of questions yet to be answered on the use and implications of this technology, the potential it has in improving the lives of amputees is undeniable.
Thank you. Are there any questions?
Here's the light microscope part we were talking about on the left. Also, the device that acts like an MRI scanner to allow for a level of control over the process, because error is always a possibility. And then the area where a limb would be inserted, which is adjustable for the arm, leg, whatever is being regenerated. And yeah.